Welcome to Physiological Psychology. I am Benjamin Miss, and this is Development and Plasticity. Let's start by thinking about what happens in our brain when we're very young. Well, there was a very particular case here with a young woman who was interviewed at about the age of 12 and was seen to be witty, charming, behaviorally normal. She was, however, missing her right eye. What was interesting about her case is, other than her right eye being missing, there wasn't really anything obviously about her that was atypical. However, at the age of 3, it was observed that she was missing the entire right hemisphere of her brain. It had simply stopped developing in utero, and they estimated that this happened around the 7th week of gestation. So, less than 2 months of development, where there's very, very little organization. Her right hemisphere just stopped developing, and there was almost nothing to it when you look at images of the brain. Now she's also missing her right eye, and we also know that the eye tissue is pretty much brain for all intents and purposes. It's the back of your eye, it has neurons, and so, but she was able to function fairly well, and again, behaviorally quite typical. Now, there's another case, Jody Miller, who around the age of 2 was diagnosed with 3, these intractable seizures. She was having seizures nearly constantly due to a rare type of encephalitis, right? Brain inflammation. And when no treatment worked, the last thing they could do was to remove half of her brain. So they removed her right hemisphere. And then there have been a lot of kind of follow-up on her. So we have quite a lot of information. If you want to look up Jody Miller, you can find a lot of information on how she progressed. Now, by the time she was like 6, 7, 8, 9, she was functioning, back to school, everything seemed pretty typical. However, her left arm never quite developed she never quite developed full use of it. And this is likely due to, remember the right hemisphere, it's called contralateral. Each hemisphere controls the opposite side of the body. The eyes are a little different. Each hemisphere of your brain controls the opposite, or is perceptive of the opposite visual field in addition to that. So that's a little bit different. But this means that they removed her right hemisphere, and she lost her left arm and left legs, kind of like that control. Very quickly, she was walking. And I think that this is due to, you kind of need to have your legs to walk, but if you're not aware, because remember, if you're missing a hemisphere, it's not just that you lose the motor control, you also lose the awareness, you lose the proprioception, you lose the sensitivity and the processing in that part of your body. So her arm, she would kind of like just ignore it, and it would take a lot of work to kind of force herself and to train herself to use it. So she did get back some use, but not of her hand and not full use. What this is getting us to is the idea of plasticity, right? So when we have this, idea, it means that the brain is changeable. This shouldn't surprise us at all. We saw this in regards to how neurons change. If we're going to store new information, we need to change the neurotransmitter balance. We need to change the number of synapses we have, the strength of those synapses, the number of receptors in those synapses. So we know the brain is changeable. We know that you are capable of vast and fantastic change. And even in a case like this, somebody might lose, for instance, their ability to speak due to a stroke. And in many cases, they can gain it back because of this plasticity, other areas taking over, but in many cases, not so much. And the idea is, the older you are, the harder it is. And this comes from Penfield and some early work of looking at children who had strokes compared to adults who had strokes or brain surgery or brain damage, and seeing the children tended to adapt better. And the reason is their brains are still developing, which means they can change, and other areas can take over. In Jody's case, other areas can take over. When we talk about development, we have to consider the anatomy. How does the brain change over time? Our abilities change over time as our brains change. There's genetic involvement, and epigenetics. And then we consider how it happens from in utero through childhood, adolescence, and even adulthood. Now, we look at the anatomy. We should have a pretty good idea of this. We've studied this pretty well. When you look at these images, you should be able to see, for instance, if I were to say frontal lobe, parietal lobe, well, each of these brains is facing a different direction, right? So toward the middle of the screen for both of the brains is the frontal lobe, and toward the edge of the screen, or lateral, if we're talking anatomy terms, we would be talking about the occipital lobe. One of these is a sagittal midline cut through the brain. The other is the left surface of the brain. And you should also, if I say things like thalamus, hypothalamus, cerebellum, you should be able to identify these things fairly well. This is important because when we talk about different brain areas, we want to have a general idea of where we are. So it's important to review this and make sure you're comfortable with these areas. Now, when we talk about genetics, we're talking about things related to DNA, right? Germ cells, genes, right? Basically like sperm, ova, fertilization. What is happening at this time? How your genetics are different, for instance, from your parents' genetics, simply in a mixing, and in addition to some mutations. Epigenetic is relating to what happens after. So what genes get turned on? And genes get turned on and off over the course of development, which means different parts of the DNA are being copied as RNA and sent to ribosomes and then making proteins that are going to have various effects. And these proteins are things like hormones. And we know that hormones are going to vary over time. Sex-linked traits are traits that are specific to the X and Y chromosomes. They don't necessarily have to do with sex, though, but they are related to that. The other ones, the autosomal traits, are the ones that are on the other chromosomes. And then when we consider and recall the words dominant and recessive, and the idea of codominance. So we think about very simple dominant and recessive. The classic example was eye color. They used to talk about that in biology classes. So if you had one gene for blue eyes and one gene for brown eyes, you would have brown eyes because brown is dominant and blue is recessive. It's not quite as simple when it comes to things like eye color, but that's the general idea. Then you have codominance. You think of things like A, B, O blood groups, where A, if you have one, let's say one parent is A and one parent is B, you could have AB as a blood type, right? Both of these genes being expressed. You could also, if someone's A or B, they could be A plus O, which is recessive. So it's an interesting combination here where O, well, it's just not expressed. We look at how we interact with the environment. And one of the things we notice right away is even as a newborn, you have influence on your environment. Right? And that sounds a little odd to us because really all you do is, cry and poop and eat. However, well, of course, sleep. However, there's different kind of types of babies. There's easy babies and difficult babies, slow to warm up. And what this means is easy babies very quickly get into a set pattern, right? Slow to warm up ones takes a little while, and then the difficult babies don't. Your sleeping is variable, your body functions are variable. And so this can have an effect on the environment because, a well-rested parent versus a non-well-rested parent. So you look at how the genetics affecting neural activity, affecting behavior affecting the environment. So we say genes don't directly affect the environment, but they do through this process, right? In the same way as adults, your impulses don't necessarily affect the environment because you do have control over your behaviors. Now let's look at some of these, early development, the process, right? So very early on, you have fertilization, right, which is happening in this, fallopian tubes as ovulation is taking place. And the egg becomes fertilized. You ovulate from the ovary into the tube. The egg becomes fertilized, and as soon as it becomes fertilized, it starts to divide. Now, this means that early development is going from the stage of the fertilized egg, which is the zygote, to the blastocyst. And the blastocyst is is part of this process and yet is not yet attached, right, to the uterine wall. So what this means is, before you're implanted, you are still having this development happening. You're still dividing into cells, between 70 and 100 cells in the blastocyst. And it's only after you implant that you start to go through the embryonic development, which means getting nutrients from the mother's body. During the stage of the blastocyst, it's just a fertilized egg that's floating through, dividing in the same way you think of like a chicken egg, right? Has all the material it needs to grow a full chicken. Human egg does not. You can start the process, but you have to do more. And what's interesting is, over this course, the vast majority of fertilized eggs, right, zygotes, and they never implant. So the vast majority of these are spontaneously aborting due to some error in implantation, some genetic issue. Something is just not complete, and we have a lot of incomplete genetic material, and so these are just going to kind of move out. The majority of miscarriages happen before the mother even is aware that fertilization has taken place. So this is what we see here in terms of the blastula. You're not getting any kind of increase in size until implantation, but you can divide quite a bit up to that point. Now, once we implant, we see there are 3 layers that are going to form, right? So the blastocyst starts folding over, and once you implant, you can start getting nutrients from the mother's body, right? And you're starting to going to be developing the embryo as well as the placenta, right? Which is going to kind of both hold nutrients and protect the baby. Similar in a way to the blood-brain barrier protects your brain and gives nutrients, and it allows nutrients to go through. So you've got the endoderm, the mesoderm, and the ectoderm, right? And those are basically just describing these layers, the endo being closest to the inside, the mesoderm being the layer above that, and the ectoderm being outside. So we look first here at the ectoderm, right? And we also can sometimes call this the neuroectoderm, or the part of it that becomes this, because the ectoderm, in addition, becomes skin and brain, which is interesting. So your skin, nails, hair, and your neurological stuff is all in this layer, right? The mesoderm is your circulatory system, lungs, your internal stuff here, but your endoderm has some of that too, including your digestive system, liver, pancreas, and lungs. So you've got these dividing into layers, and you still see it's not well differentiated. Now, we're still looking at here what's called a neural tube. This is going to form during the process of folding, where you see this neural fold happening, and then it converges and it forms this tube. This tube is going to form the brain tissue, right? And this is about 25 days. The anterior end develops into the brain, the posterior end develops in the spinal cord. So we think back to the young, to the girl who had lost, had half, where development had stopped for her right hemisphere. Well, looking here, 25 days, it's about not quite 4 weeks in. So we're seeing very little differentiation at this point, right? Around the 4th week of development, more complex structures do begin to form, but we're still talking very little. Now, a lot of organization of the brain happens in the first 2 months, right? The very, very early stage, which is the most really dangerous and fraught with errors that can happen and more kind of spontaneous abortions due to a non-viable, embryo. So we see this folding over, and we can see the layers of cells in the neural plate. We can then see the neural groove, the neural fold, and the neural tube. And you can see what this looks like. This is a cross section where you're looking at here of this kind of developing, this blob that is the very early part of development. So now we look here at how the brain is differentiating. So this neural tube is what we're looking at as it starts to differentiate. And we said the top is going to be the brain, and the posterior is going to be the spinal cord, right? And then you're going to develop your nerves off of there. So here you're looking at some terms, including the prosencephalon, the mesencephalon, and the rhombencephalon. These are the main parts, basically the fancy terms for the forebrain, midbrain, and hindbrain, right? And the hindbrain is going to form into, this is kind of simple, the pons, cerebellum, and medulla oblongata, right? Which we generally just say medulla. The midbrain is going to stay there, and then the prosencephalon, the forebrain, is going to split into the telencephalon and the diencephalon, where the telencephalon is kind of like that. When we think of brain, like the surface of the brain, that's really what we're talking about, the telencephalon, with all those nice grooves and folds and that really kind of high-level processing. The diencephalon is kind of when we think about the limbic system, right? Thalamus, hypothalamus. So we kind of think about it that way, like these really old mammalian type structures that happen very early in development. That's our diencephalon, where the telencephalon is the kind of the newer stuff. Now, here's the thing. We're still talking about how are we seeing this development? Here is our zygote, right? And then blastocyst, and then embryo. What we're looking at here is, in this image, is an embryo at 7 weeks. And we're still, looking at very much the kind of alien-looking, big head, big eyes, very, very small. We're talking very tiny developmentally, and kind of going through that process, still having like a tail and things at this point. Now, one of the things that is fairly common when a woman is pregnant is they might say, don't talk about, you don't tell people you're pregnant because so much can go wrong, right? And you're still talking about a large percentage of embryos don't make it to the fetal period because of, again, there's, you see this 1.5 centimeters, very tiny, right? 1/2 inch in size. And after 8 weeks, you technically are the fetus, but it's not like there's a hard cutoff there, right? We're still talking the first few months being at this area. Now, interestingly, there's been some kind of change in attitudes toward this, because historically it was like, well, you don't want people knowing for some various reason. But more recently, people are saying, well, why not, right? Like, if something does go wrong, this could be very, very difficult, and in some cases traumatic for the mother, and support is important. And as a culture, I think that we have not done a good job of talking about things like miscarriage and developing support for, these, pregnancies that don't go to term, which is, again, more than half. The majority of them spontaneously aborting before you're even aware. But then at this point, we're still talking a high percentage that don't quite make it for some reason or another, some chromosomal issue, some genetic issue, some other type of just combinatorial issue that happens during this process. So however you're doing, you beat the odds just by being born, right? Now, so let's look at kind of, here's the point, this kind of 2 to 8 weeks, this 7-week embryo, this is the size of the, that girl whose brain stopped developing around the 7th week. So she was about a centimeter and a half large. Like, that was basically how large she was. And so you can see very little is developed. What this means is, in her case, the reason that she, they didn't notice anything was atypical. She had full use of arms, legs, communication, was this early reorganization. And the earlier your brain reorganizes, the really the better it's going to do at reorganization. Now, there's these things called proliferation zones, right? So you've got this kind of developing embryo, this kind of growing, into the fetus, and you're producing a massive amount of neurons, right? Because this is, very, humans have a large brain comparably. So we're going to start in these proliferation zones, right, that are very kind of like deep and kind of spread outward. And there's two ways this happens. One is through passive cell displacement, and one is through what's called this inside-out pattern. Now, there's another type of glia that we don't really talk about much until we talk about development. They're called radioglia. These are basically these radiations that kind of, come out from where the brain is developing from. And the passive cell displacement is kind of like this bubbling up, like neurons kind of like, in a way, if you've ever, had dough rise, right? It's just kind of like rising or, dividing yeast are kind of pushing these things out. But for the radioglia and for this inside-out pattern, you've got these long, kind of long radiations where the neurons are actually climbing, and they're following these chemical trails that tell them where to go and where to end up. Okay, now let's see if we remember about our neural tube. The top part of the neural tube becomes the what? Prosencephalon, mesencephalon, rhomencephalon, or spinal cord. So this would be the anterior portion of the neural tube. And this is the prosencephalon, right? So the forebrain part, right? Where anterior is prosencephalon, posterior is spinal cord, and then prosencephalon, mesencephalon, rhomencephalon, spinal cord. What is the name of the fertilized cell mass that is about 70 to 100 cells that gets to this point basically right before implantation? Is it the embryo, zygote, blastocyst, or endoderm? And the answer is the blastocyst, which brings us to our next question, which is, before the fetus develops, what is the proper order of developmental stages, right? So we got embryo, blastocyst, and zygote. What is the order? What's the first, second, and third? Is it embryo, blastocyst, zygote, blastocyst, zygote, embryo, zygote, blastocyst, embryo, or embryo, zygote, blastocyst? And the answer was the third one. Zygote is the fertilized egg. Blastocyst is it starts dividing, becomes a blastocyst, and then when you implant, you get the embryo. So some interesting things about how axons grow, and there's still to this day a great deal of research being done with newts. And one of the reasons that people get excited about things like newts is, salamanders, is kind of regrowing their body parts. They're able to regrow axons. If you were to have an axon severed, right, especially in a peripheral nerve, you would probably never regain any function to whatever it was. So if you lost, for instance, nerves in your upper part of your arm, your hand would go. You would basically have no control and no ability to use that hand. So studying these is really quite interesting. One of the things that Sperry did was to basically cut the optic nerves for the eyes and then rotate them 180 degrees. The axons would then reach their target area following a gradient of chemicals, and they would reconnect. And the salamanders would be able to function. You've also had cases of removing a leg. The salamander would then grow a new one, and then they would attach the leg somewhere else on the body, reattach the leg. And that reattached leg would basically be a fifth leg for the salamander that would function fairly typically by regrowing the axons. So some very interesting work done here with how these work and what can happen. Well, axons pathfind through this process of chemical attraction, and when we see what happens with humans and with development, axons reach their targets and just start forming synapses all over the place. One of the interesting things about human development is that a very young child has more synapses, a smaller brain, but more synapses than you do. And the reason for this is these synapses are forming all over the place. The axons make the connections and form a whole bunch of synapses. Over time, then the infant is in the environment and learning and experiencing it and organizing. Remember, a newborn infant is very non-functional, right? Needs a lot of support to survive and thrive. And so through this experience, they're starting to organize their visual systems, their auditory systems, their movement, and they're getting rid of the synapses that aren't necessary. It's almost like an overwhelming amount of information that they then have to kind of sort through, and their brains have to organize. So the formation or elimination of these connections depends upon input. Are you getting a signal, and what is that doing? And if you're not getting a signal, your brain is just going to get rid of the synapses. The synapse is not useful. It's not doing anything informationally, and this is called synaptic pruning. And this process is also referred to as neural Darwinism, basically the idea being the survival of the fittest, which is a bit of a misnomer in terms of Darwin, but that's a general idea, right? The ones that you use get stronger, the ones that you don't are gone. Well, when we look at what is happening, external genitalia don't develop until around the 3rd month of development. And I know there's a lot here in the image, but the part that we care about is basically early on, right, the proto-human, the fetus has the same basic material that become the internal and external genitalia and sexual organs for males and females, which should make sense, right? The change doesn't happen, the differentiation doesn't happen until around the weeks into development, when testosterone triggers the development of the testes and produces testosterone. The testes then produce testosterone, and so this you get this gene, the SRY gene that we see here, which is then basically causing the testes to develop from these general gonads. And then you see that basically all this material for both male and female genitalia is there and starts to differentiate at that time point. And of course, there is overlap in there also. We look at the behavioral cycles, and it's also clear that during the later part of pregnancy, they tend to stabilize, which means the developing fetus at this point is going to go through sleep and wake stages that are predictable. And by the time they reach the end of pregnancy, they're similar to the sleep and wake cycles of the newborn, right? So the newborn doesn't seem to get on a real schedule for quite a while. They need a great deal of, especially in the first few months, they need a massive amount of care and support. And one of the things that they kind of mention is this, almost like we really need a year of development, but we only get about 9 to 10 months in utero. And so the rest of that year is the brain is developing, but this, you see other animals born, like newborn animals that could be walking very shortly after birth. And for humans, that's simply way too much neurological organization needs to happen before even crawling or being able to have kind of that kind of volition over what they want to do. Now, in terms of early fetal experience, what structures are available? Well, we know that the visual experience is negligible. What are you getting? You're getting, it's dark here. You might get a little bit of light change, but probably not, right? So it's very little visual developing, and they say that a newborn can focus, a very small range in front of their face, but even then there's very little comprehension, really no comprehension of what they're seeing. They're still organizing that, right? They do experience tactile stimulation through moving their hands and feet, touching, things in there, touching themselves. They develop a sense of taste and smell with the amniotic fluid, and they're also kind of developing a preference for, the parental foods, right? So what the mom is having during during her pregnancy, what she's feeding in milk shortly thereafter. The smells are very kind of already preferred. By about the 6th month of gestation, they are responding to sounds. And in relation to sounds, there is some language organization happening. Now, language doesn't really develop in a human until you really start to get this big boost of language about 1.5 years, 2 years old, right? So that's very early, is just kind of developing these patterns and coordination of the mouth in order to do speech. However, we do see that newborns, like basically right before birth, 33 to 41 weeks, this is the very end of the pregnancy, they can tell the difference between their mother or another person reading the same paragraph. And the way they study this is by measuring heart rate. They basically put a heart, can detect fetal heartbeat. And this is work that's also done with newborns, which is, how do you know if the newborn is aware of change, right? Newborns are basically a blob. They're not doing anything. So if you play for them sounds, we know that they can hear, right? They can differentiate, kind of where a sound is coming from at birth. So you measure their heartbeat. And so you play sounds for them, and if their heartbeat increases, it means they're aware of some kind of change. That's generally the way this is interpreted. The heartbeat change means they're aware of consciously some kind of change. So they're developing these kind of organizational patterns for what their native language sounds like, right? What the speech sounds like. Now let's look at this, again, incredible rapid growth of the brain. So what we're looking at here is, look first at the right side of the image where you see the mid-gestation human and the newborn human. Now the mid-gestation human, you see this brain is fairly smooth. And in rare cases where the brain never develops the sulci and gyri, you have severe developmental delay and extremely atypical development and ability to function. Now, what you see toward the end of pregnancy is this kind of wrinkling, and the wrinkling is what's increasing the surface area of the brain. This is where the really high-level processing is happening. So we're looking at, okay, the adult rhesus monkey, these are also known as macaques, very social, intelligent, old world monkey, has about the same size as a mid-gestation human. You can see it's 3 centimeter on one side. So you got kind of like a 6 centimeter, wide brain. What you see with the newborn human is the brain has increased quite a bit in size, but nothing compared to the adult brain. And remember, one of the reasons is if the newborn had a larger brain, its head would be larger and more newborns would historically die in childbirth. Not anymore because of cesarean sections. So because a newborn can be cut out, there is some evidence that we're kind of changing the head size of newborns in humans, because newborns that would historically have died because they couldn't fit through the birth canal are no longer doing so, which means we could be increasing the head size of the human population by this. So here we get the general idea of the sizes of these brains, and remember the sulci and gyri developing toward the end. Now, here is how fast this happens, how fast things happen. After birth, the brain grows by approximately 1% per day. It's increasing in size, right? Overall volume. At 3 months, this slows to about 0.4% per day. Still a lot. But you think about that 3 months, that's 90 days, okay, of growing 1% per day. So we're talking about a massive increase in size, going from this 13 ounces to likely double that in a fairly short period of time. Now, let's look at a couple things to kind of make sure we're comfortable. We remember these terms. The top part, the anterior portion of the neural tube, becomes the telencephalon, midbrain, hindbrain, or spinal cord. And this is the telencephalon. At about how many months of development will the developing fetus respond to sounds outside the womb? the funny, odd, old study from almost 100 years ago when they really tried to figure this out and looked at the idea of habituation and learning, was they honked a car horn, right, which is quite loud. And what you would see is an increase in the movement of the fetus. And if you keep honking it, eventually the fetus gets bored and is like, okay, I'm used to the stimuli. Well, getting bored because you get used to a stimuli is called habituation. And the idea is this means that learning is happening. So that's the idea of why that's kind of interesting. And the answer is, what is it, 2, 4, 5, or 6 months? The answer is 6 months. When presented with a stimulus they have not previously seen, newborn heart rate should what? Increase, decrease, or stay the same? And the answer is increase. They're interested. We should see that. We should see that in humans too. Adults. Now we're still looking at brain development, and look at how much happens. These are MRI scans. Okay, so by 3 months, look how larger that brain is compared to the 1 week old, and at 1 year. By the time a child is 2, their brain is 80% the adult size. Now there's also something really interesting here too, and very important to note, which is this. A child, a infant and child that's ignored will have a severely lowered overall size of brain. This doesn't mean you need fancy toys. You don't. You don't need stuff that lights up and sings and makes noise. You don't need a lot of stuff. When we talk about an impoverished environment, we're simply talking about an environment where there's not a lot of interaction. The most important thing is that interaction, right? And that kind of stimulation. There weren't always toys, but kids would always play with rocks and sticks and interact with the environment, right? As opposed to being isolated, stuck in a room, kind of alone. So it's really important to remember, and this is what Piaget would call the little scientist, right? The child wants to learn, right? And he was referring to older, but we see this too. Why does a newborn learn to crawl? They want to get places, right? They have this kind of drive to grow and learn. So let's look at a little more of brain development, right? We also see what's called the myelination, right? So the kind of more purpley colors here represent increased myelination, the kind of reddish yellow less. And so the idea is the brain is developing rapidly, but it's also myelinating, right? So the 2-year-old brain has 50% more synapses than an adult. So I mentioned how their brains are, smaller, but have more synapses because as these neurons are growing and developing, they're just creating all these connections with other neurons, and you eventually have to prune and get rid of these things. And the synaptic pruning is shaped by experience, interacting with the environment. By the age of 5, the brain is 90% the adult size. And you see the real big boosting, not a lot of real big boosting since then. You do see growth in puberty, you do see slow growth through, childhood. But the brain is almost completely developed by the early to mid-20s. What this also tells us is, be careful with your brain. Wear a helmet, right? I know is it still cool not to? I'm not clear on that. But you protect your brain. Also, chemicals, toxins. You got to be careful because the developing brain is much more susceptible to really permanent injury. So we're looking at, in regards to that, what are called critical periods. This is the times when input is necessary for proper organization. Some early work done with by Hubel and Wiesel, where they took cats and would kind of cover one eye and then uncover the eye after a certain period of development. Sometimes the brain would never really organize, typically, because you need that input in order to organize. There's cases of people who are effectively born blind due to cataracts or something, and the later surgery removes those issues. And what happens is they don't get vision, right? They're never able to There's an old philosophical thought experiment. What would happen if you didn't have vision and developed it later? Well, we know the answer. The answer is you would never really develop vision, right, after you go through this critical period, and that people who have these kind of operations to fix their vision often prefer to close their eyes. So much information. It's not organized, and it doesn't mean anything after some point. The brain's going to say, well, we're not using these for vision. So what does a blind person's brain do? Well, they're going to do something else. A blind person's brain is going to use those neurons that would typically process vision and process spatial information, right? Because maybe auditory stuff. We know, for instance, that people who are born blind can actually hear faster. You know how you increase the speed of YouTube videos when you're, watching lectures and stuff because you're like, all right, whatever, you're talking slow, I'm getting bored. A blind person can actually hear faster. So you can increase the sound to the point where you can no longer hear it. You can no longer understand what's being said, but they still can. Language is argued to have a critical period of about, between about that, 18 months, 2 years to about 6 or 7 years. And if you don't develop language by that time, you're going to have severe deficits in language. Someone who maybe has, hearing issues, right? Maybe they're born deaf and their family doesn't know sign, would have those kinds of issues, right? You have to have language. There's something called apoptosis, which is a programmed mechanism of cell death. Sometimes neurons dying aren't a bad thing. It's just they've served their purpose and now they're gone. And we have, remember, this idea of pruning. You want to get rid of extra synapses, synaptic pruning. There was one, autism is really complex, but there is one kind of explanation that involves synaptic pruning not happening. Too many synapses, because that would explain the overwhelming amount of information and why people with autism often feel there's too much going on and they want to withdraw. We also have this term called teratogens. And teratogens, that's a term for anything that can cause permanent damage. And it's very dependent on timing and dose. And so basically, drugs and alcohol, right? Let's say that you're a teenager and you're getting like super wasted. That's bad for your brain, right? It's also bad for your brain if you're in your 30s and you're getting blasted, but it's not as bad, right? So it's never a good idea. You're doing damage, but the younger you are, the more damage you do. So I'm not saying, once you're in your late 20s, you can start getting like really hammered. I'm definitely not saying that. What I am saying is you definitely don't want to do it when you're younger. You want to give yourself the best chance for optimal cognitive development. So teratogens include things. So looking at specifically in utero, well, what happens when the mother is taking drugs and alcohol can cause severe damage. Things like meth and ecstasy can lead to major problems with brain development, palate development. Alcohol can cause fetal alcohol syndrome, especially when coupled with smoking and malnutrition. You have a extreme, so a mother that smokes, eats poorly, and drinks alcohol is basically wild risk factors for fetal alcohol syndrome. Environmental pollutants. You think about things like pesticides. There's been some really controversial work on certain pesticides causing, issues with sex organ development in developing humans, right? It's a lot easier to see the external than the internal, like the brain stuff, but really kind of, I would say scary stuff there. Occupational hazards. Let's say you have a mother that's around toxic chemicals, right, as part of her job. That could be an issue too. We mentioned malnourishment. And then we're talking about anything that can cross the blood-brain barrier. So things that can cross the blood-brain barrier can also cross the placental barrier. All right, so you get some protection, but not to everything. So now we get into kind of more development. We look at adolescent brains, and we saw this image already. And what we're looking at is a balance between the frontal and limbic systems. These become, in like a 10-year-old, it's fairly well balanced. They tend to be pretty good at, ignoring impulses or inhibiting their impulses. But once you hit adolescence, these impulses become problematic. And the reason is you've got this increased development of the limbic system, right, amygdala, and your frontal lobe hasn't developed your ability to inhibit, because our frontal lobe is very much involved in, hey, don't do that. That's not a good idea. And so this balance is off. And so while the frontal lobe is continuing to develop, the impulse system is there, which is why you tend to see these, sensation-seeking, risk-taking behaviors of adolescence. Also the hormone changes leading to, emotional ability. So we look also at the sensitive period here. Here we're looking at, the use of drugs, sensitive period, alcohol, and you see a reduced gray matter, right, which is the really good high, processing stuff on the surface of the brain, lower brain activity, lower sustained attention, and poor working memory. So the earlier you see drug use in adolescence, the more of these issues you see. And I know that there's a difference. Not all drugs are as bad. But at this point, you're talking, it's still, you have the greatest effect at this point. You look at the things like impulsivity and short attention span of adolescence, right? Well, again, the lack of the organization, the lack of controls. And we know that full brain maturation is sometime between 20s and 30s. We're talking about the myelination. This is the maturation, not growing new neurons, but increasing the myelination. And we should remember myelination from talking about, neurons in the synapse. But remember, this is like the insulation, the fatty insulation around our neurons. So the neurons that are basically these connections, these ones sending a lot of signals, are going to have heavy myelination, right? And so the more these things are organized, the better the communication. Now we continue to age, and once you kind of hit this, point here in your mid-20s, you've really reached almost like your kind of peak. Now, it doesn't mean that it's all downhill from there. You don't actually necessarily decline for quite a while. So let's take a look at what this means, right? Your white matter organization is peaking around age 40, which means you're still developing these connections. So you're strengthening your organization until around 40, right? You're developing improved cognitive control. In a lot of ways, it's inhibition. It's the ability to delay gratification. It's the idea that, hey, if I stay up until 5 in the morning, I'm probably not going to feel so good tomorrow, or things like that. Intelligence peaks between about 25 and 60. So, you hear like, oh, this is your peak, and then you develop, you decline. Well, you're still developing. Fluid intelligence peaks around 30. Right, which tends to be the really creative years for people, which is why you see people in their 20s and early 30s, artistic, design, entertainers, really kind of developing at that point. And then cognitive decline begins between 30 and 60, and a lot of it depends on what you're doing in order to kind of maintain your cognitive abilities, right? The last areas to develop are the first to decline. The association connections within hemispheres, the temporal and frontal lobes, and the shrinking dendrites in the prefrontal cortex and hippocampus. Now, what's interesting is there is very strong evidence that you eat vegetables and whole grains and you slow your brain's decline. Dairy and meat increase your brain's decline. Interesting. I will tell you more about that in just a moment. But there is some very strong evidence that, are you a person who avoids fruits and vegetables? Are you a person who tends to eat processed foods and stuff like that, fast food, takeout, prepared foods? If you would change your diet and begin eating 4 servings of vegetable a day, you would have a mood and energy boost the equivalent of taking a powerful drug given for depression. All right, some quick questions. During development, axon growth is guided by innate axonal planning, chemical attraction, ionic attraction, or random movements. And the answer is chemical attraction. The brain fully matures by approximately which of these? 6, 16, 26, 36. And the answer is 26, but with an asterisk. Because what are we talking about? If we're talking about myelination, it's a little older. Generally, we think of a fully mature adult brain about 26. When is the brain 80% the size of the adult brain? 2 years, 6 years, 10 years, or 16 years? And the answer is 2 years. Now let me show you the evidence for what I'm talking about in regards to healthy eating. So they looked at people who were basically, varying their amounts of fruits and vegetables. And so you saw the biggest improvement for simply adding 1 portion a day, right? And this unit, this 1.33 unit of the GHQ-12 scale, this is a measure of mental health. So increasing vegetables had a clear and potent change. So going up to 7 servings a day, or 8 portions of fruits and vegetables a day, leads to a 0.24 unit increase in life satisfaction. And this is the equivalent psychological gain of going from unemployment to employment, right? And we know how hard it is for people unemployed, the amount of stress. Increasing your fruits and vegetable intake will give you that sense of well-being. That is incredible. All right, so you heard your parents say, eat your vegetables, probably, right? And, well, here's why. Other researchers looked at the foods themselves to see which ones were actually had the biggest effect on mood, the biggest kind of antidepressant effect. So here's what they did. Right, they looked at these, things that are used to treat these disorders. We know that the fruits and vegetables are better than taking supplements because what has high portions of these things? The highest scoring foods were bivalves, such as oysters, mussels, seafoods, right? You hear about fish oil, various seafoods, organ meats, which I don't think many people enjoy, and for plants, it was leafy greens, lettuce, peppers, cruciferous vegetables. I believe those are things like broccoli and Brussels sprouts. So here's the animal foods. And here's the thing. No matter how good these were, the vegetables were better. So oysters had this 56%, score here, and you really drop down quite a bit from there. Fish, good. So you look at these kind of like fish high diets tend to be pretty good, right? But not as good as this. Watercress, spinach, other greens had extremely high scores. And as you go down the list, you see even, things like broccoli and cauliflower having a higher score or as high of a score, peppers as the best seafoods. So that should be interesting to us too. Right. So a mix of these things would suggest, you have seafoods and vegetables having the highest effect on mood. So we're talking about why this is. In general, eat more fruits and vegetables, right? And then we look at, again, how do we slow decline? How do we improve our well-being? Well, they were looking at alternatives to antidepressants. Here's another study. And they found that, these certain things that exist in plants, fruits, vegetables, bee products, the flavonoids and glucosides, are used and have a very good effect on mental health and mood. They also found, and this is rather unfortunate based on what I see people tend to order from the cafeteria, sugar and fat-rich foods may increase the risk of depression. Well, what are we talking about here? Delaying gratification. We're talking about maintaining self-discipline. Extremely difficult to do, but the payoff is great. Well, I hope you enjoyed the discussion today about development and plasticity, and I look forward to future discussions in physiological psychology. Thank you.